Good day. For our next lesson, we will have the sex chromosomes. So for learning outcomes, okay, we'll just discuss the role of X and Y chromosomes in different sexual differentiation, and also the Lion hypothesis, or we have the dosage compensation. So for our outline, so we will start in the introduction first. So, okay, sex chromosomes. They are an important pair of chromosomes, of course. They have the role in determining gender, allowing reproduction as well as procreation, okay? So, sex chromosomes and haploides, they are the most common chromosome abnormalities seen in infants, children, as well as adults, okay? Here, let's focus our attention in this picture here. This is a female. They have two X chromosomes. This is a male having X chromosome along with a Y chromosome. Okay, the difference in the chromosomal constitution of the males and the females is a chromosomal mechanism for determining sex at the time of fertilization. Okay, so as you can see here, the egg cell contains an X chromosome, okay, and the sperm cell contains X chromosome, and the rest will have the Y chromosome, okay. So during fertilization, there is an X bearing egg by an X bearing sperm here, resulting by a female, an XX female child, and the fertilization by a Y bearing sperm and the X-bearing eggs will have to develop a male or an XY child. Okay, so this is the result of a crisscross pattern of inheritance of the X chromosome, in which a male receives his X chromosome from his mother and transmit it only to his daughter. Okay, so for the next, so we will discuss the sexual differentiation of X and Y chromosomes. So what is the role of X and Y chromosomes in sexual differentiation? First, we have this genetic sex. It is established at the time of fertilization and is dependent on whether an X or Y bearing sperm fertilizes the X bearing egg. Okay, so the type of gonads that develop is determined by the sex chromosome complement. If XX ovaries, if XY you have the testes. So before the 7th week of the embryonic life, okay, the gonads of these both genders appear identical, but because of this gene, which is the testis determination factor, and under the influence of this Y chromosome, the immature gonad becomes a testis for male. If there is an absence of this Y chromosome, the gonad will differentiate into an ovaries for female. So this is what we call the phenotypic sex or the anatomical sex. It refers actually on the appearance of the external genitalia, and in some disorders might not correspond to the gonadal sex. Example of this disorder, we have the true hermaphrodites. Okay, it will be discussed in the other module. So next, for the next topic, we have the X chromosome inactivation. Actually, there are thousands of genes in X chromosome, but only few present in the Y chromosomes. So the fact that males can survive with only one chromosome, female has two, um, is because of these what we call the dosage compensation concept, which is termed also which is Lyon hypothesis, and I will discuss it more further at the end of these topics. Okay, so in in females, only one chromosome is active because of X inactivation that occurs early in embryonic life, like 3 days after fertilization, and is completed by the end of the first week of development. So the inactivation is random, meaning is either maternal or paternal X can be inactivated. Okay. Also, the inactivation X has properties characteristics of heterochromatin. So, with late DNA replication in the S phase of the cell cycle and remaining condensed, which is during the interphase. Okay. So, a gene that can control this process is called the X inactive specific transcript or cyst. It is located at the X inactivation center at band XQ13. So only the inactive X this can express this gene, and this gene codes for the cyst RNA, which appears to paint the inactive X chromosome and is once responsible for an inactivation. Okay. Also, let's note that the evidence for the existence of this inactive X is because of the observation of the Barr bodies. Okay. Barr bodies are a dark staining chromatin body that is present in one copy in normal females, and males have no Barr bodies. Okay. Um, Currently, in this cytogenetic field, the techniques for detecting this inactive X is based of the fact that it is late replicating, and it involves the use of as I think it's bromodeoxyuridine. Okay, and now in current techniques, people use molecular techniques, which is a very it is I mean is very popular nowadays. Okay, so for the next topic we have the pseudo autosomal regions. Okay, so this is actually a distal regions of the short arm of the X as well as the Y chromosome here. So, okay, so they contain similar DNA sequences. Okay, and during meiosis in male, an XY bivalent is formed, and there is crossing over that occurs between the autosomes, and it is this why we call it the pseudo-autosomal or PAR1 here, example, PAR1, and it has been observed to associate with proven recombination events. Okay, we also have here PAR2 here, which is on the distal ends of the XQ or the long arm, and the YQ or the short, I mean the long arm of Y. Okay, so for the next topic, we have the Y chromosomes. Let's focus here. Okay, so this is made up of different regions. This include we have the pseudo-autosomal regions. Here you have par 1 in the short arm. You have par 2 in the long arm. Okay, they also have heterochromatic region here in the long arm, and we have the male specific region of the Y. We call it MSY, which is located here between par 1 and par 2. It has 78 protein coding genes, and it encodes 27 distinct proteins. Okay, so actually 12 of these MSY genes are expressed in many organs in our body, but only 11 are expressed in the testes of the males. Okay, so next, to continue this one, we have, okay, I've already said this, the testis determination factor. Okay, this one that leads the differentiation of the gonads into testes. It is actually located in the short arm of the Y chromosome. So this TDF is mapped by molecular analysis of a sex-reversed patient. So, and the gene, which is the SRY, was identified. So it is the sex-determining region Y. Actually, it is located the short arm of the Y chromosome, of course, the band P11.3 in the MSY region. So the Y chromosome is also a heterochromatic region on its long arm. It has this region in its long arm, and the length of this region is constant from one generation to the next. Okay, so we have a gene controlling spermatogenesis. We call it the azoospermia factor. Okay, and is located in the YQ11 region, 11.23 here, the AZF. Okay, we also have a locus which is for the susceptibility of this gonadoblastoma on the Y chromosome, and it is giving high incidence of gonadoblastoma in females. And the deletion mapping localized at this gene which is near to the centromere, and it has raised the possibility of a multiple gonadoblastoma loci dispersed on this chromosome. So we have the lion hypothesis and the dosage compensation, which is the last topic for learning outcomes, and this will also explain the X inactivation of the females. And so for lion hypothesis and dosage compensation, okay, as I discussed kanina, you have the male and female sex determination, and there's a problem of the dosage of the genes of the X chromosome because females have 2 copies and males have only 1. So question, bakit yung Y chromosome walang problema, and we will not do dosage compensation? It is because Y chromosome is largely heterochromatic and it carries only a few genes. Okay, so in most organisms, so a mechanism of dosage compensation has evolved in which the unequal dosage in the gender is corrected by increasing the activity of the genes in the X chromosomes in males, or reducing the activity of genes in the X chromosomes in females. Okay, so the somatic nuclei of a normal female, it contain a highly condensed mass of chromatin. We call it the Bohr bodies. Okay, so this after the one who discovered this one, it is Murray Bohr. Okay, and also take note that it's not found in the nuclei of the normal male cells. Okay, and in 1961 you have the Mary Lyon and Lillian Russell. Okay, they expanded this concept and now they call it the Lyon hypothesis. Okay, so this hypothesis proposed the following. We have the Bohr body is highly condensed, okay, and genetically inactive X chromosome. So, meaning it become lionized in the process in lionization. So this leaves a single X chromosome that is equivalent to the single X chromosome of the male. Okay. The second one is that the X chromosome that is inactivated is randomly chosen from the maternally derived and paternally derived X chromosomes in a process that is independent from cell to cell. So, but Once a maternal or paternal X chromosome is inactivated in a cell, all the descendants of that cell inherit the inactivation pattern. Okay, so next here is, so that's it. Thank you. And the next topic or the next module that I will discuss is kind of correlated with this. It is the the sex chromosome of abnormalities. Okay, so thank you for listening. Here are the references.